Thank you, Dr. Sudirman. Hi, everybody. I will introduce our panelists in session 3 today. Yes, the first one, I think Dr. Anicia already introduced Ian Nash, and nicely. And then the second panelist will be Sylvain Faugernon, a professor and the president of the Chilean Phycological Society. His research is combined ecological experiments and population genetics. Our next panelist will be Juliet Brody, a merit researcher in phycology from Natural History Museum in London. She is an international expert in seaweed with over 35 years experience. She focused on seaweed in a time of rapid environmental change, including a citizen science seaweed program. Next panelist. Next panelist is Jonas Collin. He is a researcher at the Station Biologique de Roscoff, France, with a research interest focused on physiology, ecophysiology, and genomics of intertidal seaweeds. Our next panelist will be Simon Davis, founder and managing director of Seedling, an environmental and social impact seaweed biotechnology company. He's a firm believer that responsible business is the key driver to creating lasting sustainability, and is a passionate advocate for the pivotal role that seaweed will play in generating novel products for our future, whilst combating carbon and restoring abundance to our oceans. Our next panelist will be Peik Im Lim, a professor and deputy director of Institute of Ocean and Earth Sciences, University of Malaya, a founder member and the current secretary of the Consortium of Southeast Asian Seaweed Taxonomy. She is involved in multidisciplinary approach, is adopted to address key issues. And she is involved in the Global Seaweed Star program. Next panelist will be Michael Y. Roleda, a professor at the Marine Science Institute, University of Philippines, Diliman, Quezon City. Experience with tropical, cold, temperate, and polar seaweed. Current applied research collaborations, including using seaweed for food, feed, biofuel, and biopackaging applications. The next panelist will be Zanny Langford, Research Fellow in Rural Development University of Queensland, a social scientist and economist with an interest in land, food, and rural livelihood in Indonesia, Northern Australia, and the Pacific. Next will be Anicia Hurtado, who's already given a welcome speech early in these sessions. She's a chair of the Integrated Services for the Development of Aquaculture and Fisheries. Currently, the developing countries partner for Philippines for the Global Seaweed Star project. I think all have, and I hand it back to Dr. Sudirman to start the Q&A session. Please, Sudirman.
Thank you very much, Dr. Inaya, for introducing all panelists in our sessions. Before we facilitate further discussions, I would like to once again express our gratitude to all speakers who come from different regions, from Indonesia to the Philippines and Malaysia, and then across Arafuran Sea to Australia, and also to Latin America in Chile, and then to Europe in UK and also France. Especially those who are attending our current webinar at odds time, we particularly express our gratitude. The different time zone will, of course, barriers in the current approach using digital technologies for our discussions. But COVID-19 pandemic that we all face at the moment also facilitate not just a crisis, but also opportunities to push us to use technologies in facilitating academic and professional discussions such as our current event. So we are going to allocate time to discuss with all of our panels, and their presentations, as we all know, have been pre-recorded, and this opportunity will be asked for all of panelists and audience to be engaged in better understanding the problems. So I would like to start with our panelists, Dr. Sylvain in Chile. Hi, Dr. Sylvain.
Hi.
Yeah. So, yeah, of course, there are a huge time difference between Indonesia and Chile, and we really appreciate your availability and also your enthusiasms to attend our sessions. And there are already several questions asked by our participants. One of them from Alan Critchley, asking that if you had to select one restorative actions, what would be your top piece of advice as where to start for the greatest impact?
So thank you, Alan, for this question. It's a complicated question to answer because there are many problems to solve, and there is a lot of things we still don't fully understand about seaweed production. In the case of the Gracilaria production in Chile, we have learned at least that there is a major issue with the amount of genetic diversity in Chile. There was, before cultivation, there was a strong effect of over-harvesting that removed most of the genetic diversity. And now, if we combine this with the current cultivation practice that is based on clonal propagation, then you have very homogeneous farms in terms of genetic diversity. And this is really one of the main factors that is promoting the decay in the quality of the production, the invasiveness of pathogens and epiphytes. And so probably we need to learn to develop some better strategies to manage this genetic diversity through breeding programs or other cultivation practices that could use better the natural diversity available, for example. So that's probably one of the actions I would try to promote.
Yeah, thank you, Dr. Sylvain. And another follow-up questions from our audience is asking why diploid populations is more preferred over haploid for farming, and does it have any genetic advantage?
So in Gracilaria farming in Chile, which is done by clonal propagation, so the people harvest the biomass and use part of the biomass just to restock the farm. we have found that most of the farms is completely dominated by diploids. And actually, we don't really know why. The farmers cannot differentiate diploids from haploids, the sporophytes from the gametophytes. So they are not actively selecting for that. There are no known differences in terms of quality of the agar. And when we try to measure some physiological aspects, for example, we know in the laboratory that the sporophytes grow a little bit faster than the gametophytes. But when we look at natural populations, for example, females grow at least twice as fast as diploids. So we don't really explain why diploids are so much successful in the farms. It could be from some kind of advantage, like the hybrid vigor, the heterosis that we see quite often in plants, in plant cultivation. So that means that you have some genetic properties of the individuals that produce like a kind of vigor. But so far, our experiments have failed to demonstrate this existence. So it is something that we don't understand. It is one of the things that we don't understand, actually.
Yeah, very insightful elaborations. And one more questions for Dr. Sylvain before we move to the next panelist. One of our participants asking about what farmers do during an aphidside attack at the moment, and how do they find sources of aphid-free biomass to restart cultivations?
That's a very good question. So, what do? Usually, they they stop producing when the price that they get for the biomass is getting too low because the quality of the biomass is too bad. For example, when there is too much epiphyte in the gracilaria production, then a large part of the biomass is not gracilaria, it's something else. Usually many different species are epiphyting the gracilaria. So the quality is decaying, and then the producers have a lower income from selling their production. So that stimulates them to remove the farm and eventually start from scratch, from new plants that they buy. But where do they buy this new material? Usually from the neighboring farms. There are not, in Chile, there is no company or organization producing clean seaweeds, clean and healthy seaweeds that was mentioning Ian in his presentation. There is a strong need of clean, healthy, secure production of juvenile plants to be used for setting new farms. This doesn't exist. So they buy from the neighbors. They have for 1, 2, or 3 years of really relatively good production, and quite often after that, the quality starts decaying and they start over. So it's not really a highly productive way of managing the production, I think.
Thank you once again, Dr. Sylvain from Chile, addressing 3 questions. Perhaps you are going back to you for more questions after other panelists. We are now going to move from Chile to London and invite our colleague, Professor Judith Brody from National History Museum, who presented in the pre-recorded presentations about the microbiome and holobiont lesson learned from one carcinophyte, chondrus. And we already had some questions for you. And one of them is from a participant asking, how easy are the technologies required by microbiome studies transferred to the field? And can countries affected by imbalances in microbiome undertake their essays? Professor Vori, time is yours.
Yeah, thank you very much. Yeah, that's a question that's been exercising my mind for a while, and I think that is at the moment that's tricky. I think what we need to do is to understand very well the whole process of what goes on over the period of time that perhaps plants are grown, either in culture or in the wild, and then try to really figure out what's happening with the plants and the microbiome, what is affecting the plants through the microbiome and vice versa, and all of that. But ultimately, you know, these technologies are getting cheaper, they're getting easier. We're developing more protocols, and I can't think of a timeline, but I would like to think that down the line, you know, we can develop some tools and some technology that's cheap to use, that's simple to use, that takes this sort of whole understanding of the holobiome, but perhaps simplifies it down to some things that are practical in the field and that can be used. But at the moment, that, in my head anyway, others may know better, this is all a bit hypothetical, and we've got a lot to learn. But I do think we can learn a lot. And, you know, for those of you that saw the talk, you'll see, you know, how important it is to really know everything that's going on in the life history, the life cycle of all these plants, whether they be cultured or whether they be in the field.
Yeah, thank you. And as you all know, microbiome topic are now increasingly become the focus of research in many different fields, and including in seaweed. And another questions, and this is asked by Dr. Nash, about your thoughts on the phenomenon of the ripening, or the so-called the Nash effect, how it applies to ecomatoids and other tropical red seaweeds, and how it may impact microbiome, epiphyte, and endophyte phenomenon.
Sorry, I missed that bit, the bit about the effect. Could you just say that again?
Yeah, the Nash effect, during which thallus color change and presumed to be related to nitrogen availability.
Oh, okay. Yeah. I don't know I don't know the answer to that question. We haven't looked specifically at nitrogen availability, but I think that is going to be something which it will be another factor affecting the microbiome. I mean, one of the things that we are doing a lot of work on, one of my postdocs who generated the microbiome data, Georgia Ward, she's been looking very closely with our global seaweed partners in Malaysia and the Philippines and Tanzania at how the microbiome changes in relation to the eye size, and trying to sort of figure out exactly what's going on there, for example. So I don't know the answers to those questions, but I think that they will in time all become apparent, and these are areas that we need to focus on more specifically. At the moment, what we've been trying to do is to sort of get an overview and to work out the methodology. And I think now that we've got that a bit more sorted, we can start to sort of delve more into these questions, and then again try to find some solutions.
Yeah, thank you, Professor Brody. There are, of course, several unknown territories in this field that require further studies.
Yeah.
And one more questions for you before we move to the next panelist, and perhaps you can go back later if the time allows. One of the participants asked about prebiotics. And as we know, that prebiotics are often used to influence microbial communities. Do you think this is possible with seaweeds? And similarly, prebiotics are often used too for human and animals' digestive tract. What chance do you think is there of introduced bacteria which may change populations or bacteria which benefit the crops of seaweeds?
Well, I think, I mean, there are people who know more about this than me, but I think already people are using seaweed extracts to prepare the juveniles before they're put out into the field and so on, to try and improve their chances. So already people are doing that. I think some of the interesting areas here that I've thought about, perhaps a little bit separate to the prebiotics, is thinking about one of the species I mentioned very briefly in the talk. So this is the group of the Osmundias and the Laurentias. You know, they have these secondary terpenes, and I think those are very interesting, perhaps in relation to thinking about those in terms of maybe control within farms. So I think there's sort of some very interesting angles, like the prebiotics, and I did wonder as I was doing this work with chondrus, whether that has the potential to produce something that could be used as a prebiotic. I don't know, years and years ago, I did some experiments in the lab, and it does have an antimicrobial effect, and that probably is linked to the microbiome. But I think these are all areas where we need to invest in a lot of work. And there's actually one more point I would just like to make with all of this, is that you know, one of the things we have to do to be able to do any of this, we have to think very, very carefully about conservation, both of wild stocks, in situ and ex situ. And this picks up again with the things that Ian was talking about. You know, we have to secure what's in the wild, and a lot of this is disappearing, and there needs to be a concerted global effort for seaweed conservation for the future, I think, of the industry in many ways. And that's both in the field and I think outside the field. So I think that's the point I want to make. There's many, many angles to this, but people are welcome to put stuff in the chat. And I've got lots of ideas, but I don't know all the answers at this stage. I need people to work on that.
Thank you very much, Professor Brody, for enlightening us with some of the latest development in relation to microbiome, and also once again our appreciation for your time. And you mentioned previously about the soccer or the football team, and I just saw my neighbors here around my house who are the supporters of England, and some of them also Italy. So we are waiting for the very exciting game in the next few days. Thank you once again, and we are going to move to Dr. Jonas Cullen.
Yes, I'm here. Do you hear me?
Yeah, Jonas, we can hear you.
Okay, good. I hear you also.
Yeah, thank you. Just unmute. Thank you very much, Dr. Cullen, for joining us. And as many of us have seen your pre-recorded presentations entitled From One Carrageenapite to Another, Insight in Ecomatoids from Studies on Chondrus crispus. And Professor Cullen, there are several questions that we managed to collect, and one of them is a questions that asking you to outline your experience with stress ecomatoids and the evolutions of peroxide. Is this a defensive mechanism? And if so, how does it damage the crops?
So yes, I think it's a response to any infection. A lot of the experiments I did was actually trying to see the interaction between stress and production of hydrogen peroxide. And I think it's been shown by myself and others that these stress responses are very important for the seaweeds to protect themselves. But similar to almost any organism, including us, there has to be a balance between the defense mechanism and the capacity of the organism to tolerate their own defense. So it has been suggested, for example, that ice is possibly created by a little bit too active defense system. So in this case, ice should be produced when some kind of stress induced a too high production of peroxides, and this, instead of curing the disease or create a defense, is actually too much.
It's killing itself.
It's killing itself. So
it is killing itself.
I tried to do experiments in Sweden with tropical eucumas, and they died. And I think the main reason was that instead of being in the sea, they were in a 10-liter flask in the lab, and then their defense mechanism was just too strong.
Thank you, Mr. Colin, and excuse the noise from my side. And one more questions from the audience asking that from your whole organism genome work, such as with Ectocarpus and Chondrus, if informations were freely available for the ecomatoids, what do you think would be the biggest benefits to the wider community of seaweed growers and phyconomists? Do you see these informations can help to solve issues that have been outlined at this workshop?
I would answer yes and no, in the sense that I think a lot of the information is freely available. I don't see any real use today directly to use for eukaryotes. However, we need to go further when we work with seaweeds. We really need to get better methods for strain selection. We need to be able to do crossings. We need to do plant breeding. And I think when we can do this, the availability of genomes and genome information will be an enormous advantage when we try to create better breeds of seaweeds. So today, no. In 10 years, in 15 years, yes. I can't hear you.
We can't hear you, Sudirman.
Okay, yeah, thank you. Thank you, Professor Colin. And one follow-up questions from Dr. Flower Mansuya to elaborate further of what you have said. And she informed us that she also tried culturing ecomatoids and in tanks in Israel, but they produced the peroxides that kill them. Do you have some response to Dr. Mansuya questions?
Yes, I failed to grow eucuma in the lab, but I've seen people that have done it. They have typically added quite a lot of antibiotics. And I think if you want to grow eucuma in the lab or in small volumes, you need probably to control the bacterial growth to reduce the defense mechanism. So I think according to the literature, and my guessing is that you need to control the bacterial growth in your cultivations.
Okay, yeah, thank you for the elaborations. And one more questions before we move to the next panelist, Dr. Simon Davis. One participant asked that he or she found in your slide on carrageenan biochemistry, and it appears that the cells can naturally convert lambda carrageenan into kappa carrageenan. Conventionally, this is done using alkali. Could this be replaced using enzyme, for example, in the future?
I mean, there is a willingness in industry and to use more green or blue technologies to do chemical reaction to produce your product. Today, I doubt very much that this would be economical. In the future, I don't know. Treatment with alkali is relatively inexpensive. So maybe in the future, probably not today, probably not in the near future.
But there are some possibilities.
There are, I think it could be done.
Okay, yeah. Okay, thank you once again, Professor Colin. And perhaps you are going back to you if time allows after facilitating time to our next panelists. And now we are going to discuss with Dr. Simon Davis. Dr. Davis.
Hello, Dr. Sudirman. It's wonderful to be here.
Thank you. Thank you. And with Dr. Inaya, we and the team here in Makassar, Hasanuddin University, we also collect some questions specifically asked for you. And one of them is about your comment on the longevity of carbon in the system as captured by cultivated seaweed. What would be the end use of the biomass if used to mop up nitrogen, presumably from anthropogenic sources? And this is from, again, from one questions from Dr. Alan.
Thank you. Nitrogen and carbon are really two different aspects to look at in terms of using seaweed to capture nitrogen and carbon. Starting with carbon, because I think there's a lot more known about it and it's a lot bigger push. There's three mechanisms discussed so far. The first of seaweed sloughing off its skin, so to speak, as part of its growing process, and that could be classified as carbon captured. The second being in the process of farming, small pieces of seaweed could be fallen off as detritus, and that's absorbed into the sediment bed. And then the last is models of open ocean seaweed farming where biomass is sunk to the bottom of the ocean, below 1,000 meters, where it's in theory sequestered. And at this stage, it's theory. But there's increasing work in this area, particularly by there's a project led by Oceans 2050, which was a study done with seaweed farms around the world, and there should be data published on that later this year. That's regarding carbon sequestered by seaweed farms. So that's really, really interesting. That will be a good first step, and I expect that there'll be more studies following that. And the hope is eventually we can have existing seaweed farms that, as well as growing seaweed, we can also classify them as carbon sequesters, which would be a wonderful opportunity for seaweed farming communities to earn extra revenue whilst doing the same activities. But as I said, the first step from that will come in later this year, and then I feel it will grow after that. In terms of nitrogen, well, that's a lot, further behind in the development stage, and that's predominantly because there's more push behind carbon politically. There's been a first step in Australia about having what's called nitrogen credits. Having for the Great Barrier Reef, there's a credit scheme where nitrogen polluters, say for example, aquaculture polluters or farmers who over-fertilize their land and can offset their nitrogen. It would be wonderful to see that come into the coral triangle, because there's no doubt there is a significant amount of nitrogen pollution. However, I think due to the lack of political will, there's a problem of enforcement, and I foresee that's going to take a lot longer to gather momentum. So in terms of nitrogen, I think we've still got a lot further to go, but it's definitely possible. But carbon's very much on the horizon.
Okay, yeah, thank you. And some questions they could be still related to the previous questions asked by one of our colleagues from Pakistan, and Ghazalabad. And he or she mentions that in your presentations, you show that alga species such as ulva and caulerpa are the future species in this sector. Yes, perhaps this is there's a growing species also grow on the rock and rapidly in Pakistan, but actually it is neglected site in this particular country. Could you suggest how people in Pakistan can convince the public that these are sea vegetables and that we can use them in our daily life or to feed our animals? And as people there try to use sargassum as fertilizers of the growth of wheat and got good result, but having no source to make a pilot plan. How do you think these possibilities can be enhanced?
Unfortunately, I haven't seen the phycology scene in Pakistan, and I haven't certainly been to the coastline and seen what's growing, so I can't entirely comment with confidence. What I would just encourage them is to look at what models have worked in other countries. So with caulerpa, it's traditionally used in the Southeast Asia region as a food, as a salad. With ulva, there's been uses as animal feed supplements, as fertilizers as well, also in bioremediation of aquaculture. Sargassum is a bit more tricky in terms of monetizing that. But there are examples, from other uses of sargassum that, from countries like Indonesia, and then growing focus in the Caribbean with their huge sargassum issues. So I would encourage them to look at examples from other countries that are already using these particular species. But in more of a long-term aspect, there's opportunities in these particularly new species, ulva, caulerpa, looking for high-value compounds. And that's more of a biotech play. It's going to take time. And there's opportunities in that as well, but it's more of a longer term, high investment project.
Okay, thank you. And one more questions from Dr. Nesh. He mentions that at the moment there is tremendous buzz about seaweed, and there are many meetings, but it seems to him that there is no way to speed up the long slope of R&D and testing and certifications and miasma of red tape that required for so many likely seaweed applications. What are your thoughts on how such rapid development can be materialized?
Yeah, I completely empathize with Ian's feelings about the potential length of time this could take, but I see it that if you look at the trajectory of previous development of, say, the last 2 decades in tropical seaweeds, and if we continue that trajectory, yes, we are looking at another 1 or 2 decades. But I would also point out that in the last particularly 5 years, there's been a lot more innovation and development in this space. And You know, I see that we're approaching a tipping point where, you know, where innovation will speed up a lot faster, and it will trickle down into, you know, real in-farm advances. I see Kappa-ficus, you know, emerging from the box of being purely for hydrocolloids, which it has been for the last, you know, 40, 50 years. And it's now, you know, breaking into new markets. And that will be the big pull as market demand pulls greater demand and greater innovation in the field by using Kappa-ficus and other euchematoids in different uses. Whether it be, you know, feed, whether it's bioplastics, fuel, you know, all the different uses. But of course, you know, there still is a huge burden of R&D that needs to happen. And the question also is, where will this come from? You know, I think the biggest challenge is the countries where euchematoid farming takes place, you know, Indonesia, Philippines, Malaysia, Tanzania. There isn't an abundance of government funding to fund the R&D. It's very different to say the scene in Europe or North America, where there's a lot more funding, but a lot less farming. So there's a lot of challenges for, you know, how this funding should take place. I see, you know, obviously we really hope that we can get funding, but we should not wait. We can't just wait for that to happen because we could be waiting a long time. I see opportunities for new entrepreneurial companies moving into this space, and that's already happening now, and that will continue to snowball. And lastly, I see opportunities for existing processing companies to be more intimately linked with farmers. At the moment, there's a great disconnect between processors and farmers. You know, it's very opaque. And that means that processors have got next to zero incentive to invest in innovations in seaweed farming. And so there's very little innovation trickling down to the farmers themselves because the processors, the buyers, don't have an incentive for an improvement. With increasing deeper relationships between farmers and processors, there will be a greater incentive. Now, key to both of this, both the entrepreneurial companies and processors investing, comes down to protection of IP. And that is definitely an issue. If there's going to be any sort of investment from industry companies, there needs to be greater protection of IP in the countries in which we operate, because otherwise they won't invest. And that is absolutely critical. And I see that as a problem that needs to be addressed.
Yeah. Yeah, one request for you, Dr. Simon, from Dr. Alan, to provide a link in relations to the carbon sequestration report that you had just mentioned. It's going to be very useful if you can provide a link about that report.
Yeah, so as I said, the data will be out later this year, so we don't have a report at the moment. I'd encourage people to look at the work of Carlos Duarte.
Okay.
But I can share a link to the website of the group. Happy to do that.
That's great. And one last question before we move to the next panelist. On the more optimistic side, one of our participants asked if once everything is in place, how many new species or cultivars of tropical seaweeds do you think could be brought into phyconomic practice? And what are the most attractive present and future markets to produce the new biomass for?
Wow, that's a wonderful question, and honestly, I've got no idea. I think there's going to be tremendous diversity needed, particularly as we start to innovate in new product uses for euchematoids beyond hydrocolloids. And particularly as we start to dive deeper into gene environmental interaction. And there's no doubt that certain varieties will perform better in different locations or different seasons. But there needs to be a lot more research into this area. And from that research will come greater opportunities in different cultivars.
Thank you very much once again. And if time allows, we still have several questions for you after facilitating discussions with other panelists. Thank you once again. And now we are moving to have discussions with Professor Paik Im Lim. And many of us had watched the pre-recorded presentations on the overview of molecular markers for identifications, taxonomy, and phylogenetic study of ecomatoids. Professor Lim, we also have several questions from the audience for you. One of them is asking about the possibility to identify disease resistance through molecular markers. Can these techniques be employed, and how do you think the prospect of these techniques?
Okay, thank you, Professor Surdeman, for chairing the session and also for the questions. Okay, so as regards about using molecular marker to identify disease resistance, I would say at this stage, we do not have enough data or information to design a molecular marker that will be able to help to identify with regards about disease resistance. So further research need to be carried out. Firstly, we have to identify what are the genes related to disease resistance that we want to look into. Probably what are the key genes, because like, for example, for eukaryotes, when we talk about disease, we always talk about ice. So what are the key gene that will be expressed if they are infected by ice, or we can have a early warning system. So we need to have further R&D before we can design molecular marker to allow quick detection or early detection.
Yeah. And one related questions asked by our participants in relations to the transferability of the technology or methods. Do you think these can be relatively easily transferred into other countries with poorer or more limited facilities?
Okay, thank you for the questions. Okay, I would say that molecular identification is something relative simple technology that you can easily transfer, and also the capacity building can be built out. So I mean, for example, through Global Seaweed Star program, our institute, we have actually accepting students from Mataram University to come into laboratory, and we teach them how to use this molecular tool for identification. But at the same time, I also understand, because some of the cultivation farm is located as quite, especially in Southeast Asia, some of it at very rural area that access, even sometimes basic electricity might be a problem. So I mean, in order to help the industry, of course, a province or national level center of excellence will help to build up, but as well as local university. But the thing is, it is not a difficult technology. Yeah, it's possible to be built up. Yeah.
Yeah, that's great to hear. The possibility for technology transfer. And one question particularly addressing Malaysian context. Many of us know that Malaysia's seaweed industry went into a period of declining. And based on the molecular work you are currently doing, what do you think are the best strategies to recover or to revive and in fact can be valuable to increase productions in the country?
Okay, thank you for the questions. Okay, so I would say that when we talk about the decline production as regards about seaweed in Malaysia, there are many factors or several reasons that I won't say that molecular marker or molecular methods will be able to solve the whole problem, because we know there's a few problems. I mean, we know that when you increase the cultivation area, you can increase the production. But another things that I think that when we talk about molecular ID that can help is actually using molecular tool, it can help to be used for the selection of the best seedling that can be used for growing, such as fast grow rate, pest, and pathogen free. Because if you were to look into, because in year 2014, our group published a paper that's on the genetic diversity of the euchematode in the Southeast Asia country. And based on the study, it shows that the three main species that we are cultivating for the region, like Kappa ficus avrazia, Kappa ficus stratus, and Euchema denticulatum is very, very low as regards about genetic diversity. So if you were to trace back on the history of domestication of euchematode, we know that seedlings are being transferred out from Philippines or Southeast Asia region to different parts. If you were to look to the time of the size of the Kappa ficus avrazia, for example, is very huge compared to currently what we have. And the main reason is we depend on only vegetative cultivation. We know that if we are using the same mother stock that continue to vegetatively culture, so with time, the quality will deteriorate. So we need to find, through molecular tools to identify and to find from the wild population, as well as to look into the genetic diversity. In that case, we can introduce more diversified seedlings for cultivation, and this will be healthy. So, and also less susceptible to any outbreak of disease. Okay, so I hope this will somehow to help. So another potential aspect that was not included in my talk is using transcriptomic to help to identify relevant genes related to stress. We know that temperature increase have been one of the, with the global climate change is one of the challenge. So our laboratory, we are studying on the identify the strain of species that can adapt to higher temperature. So at the same time, we also use transcriptomic tool to identify what are the relevant genes that's been identified. So these genes are potentially can be used as a probe, but this will take some time before we can share the result of find. And I think if there's a concerted effort among the country to pull out the strength and to look into different aspects, I think that will greatly help the industry. Okay, thank you.
Yeah. Thank you, Professor Lim. And one last question related to the potential for future commercial aspect. And this participant asked if new species should be considered, and can molecular markers can help your ID species of interest that can become commercial success in the future?
Okay, thank you for the question. So I would say that molecular techniques or molecular ID is a tool that assists us to confirm as regards about the genetic composition. So as we know that seaweed are very plastic in nature, the morphology are quite easily changed according to the environment. And we can see already in Kappa ficus and Euchema species that when we move them into different environment, their color might change, their morphology might change. So in this case, the molecular marker is needed. And then, even for when we describing the new species or to identify whether this species is the same species that we have planted after many, many decades, or this is a new, we need a tool to we need molecular ID to help to confirm whether they are new or old species, existing species. So, and as I mentioned previous question, molecular ID will help to assist the best seedling that potentially can be cultured, and that will indirectly contribute to the success of commercialization. It won't be directly contribute. Okay, so this is what I think. Okay, thank you for the question.
Yeah. Thank you very much, Professor Lin, for the further elaborations from the questions asked by our audience. And now from Malaysia, we are going to move to the Philippines with Dr. Michael Roleda. Dr. Roleda, can you hear us?
Yes, I could hear you. Good afternoon. I was muted.
Okay, good afternoon.
Unmuted, so, yes.
Yeah, your presentations on wild Capapicus populations and selective breeding as sources of new cultivars also attract numerous questions, and I would like to choose some of them and ask your further elaborations. One of the question is if it is indeed your presentation is indeed very informative and with considerable potential to improve the cultivars of ecomatoids, whether the strains selected for breeding were homozygous for specific trait. If so, what is the trait? This is a question asked by one of our participants. Could you please respond to that?
Okay, thank you for that question. So the trait that we are selecting is basically a high growth rate, and that is our preliminary determinant. And then when we observe a high growing novel cultivar, then we also try the biomass and let our collaborator analyze the carrageenan yield and quality. So in the hatchery, we first select the highest growing cultivar, and then thereafter it's the time that we look into the chemistry. Thank you.
Yeah, thank you for the clarifications, Dr. Roleda. And questions from participants, Kelvin Zhou Kang Ong, asking about how does the inductions of germinations and sporulations of the seaweed is being done, and how long does a seaweed take to finish its life cycle?
Okay, that's a very good question and might take a long time to answer. Okay, first, we could induce spore release. So spore release could either be induced by desiccating the sample for a certain period of time and then put them back in water. But in our case, we do the natural sporulation technique. No? So what we did is just cut the piece of the thallus that there is the cystocarp, or when it's tetrasporic, the piece of the thallus that we see the tetrasporangial nematotia. Cut it in 1 centimeter length, and then disinfect it, and then just put it on top of a glass slide, and we wait like a couple of days to let the spore naturally release. So we look at it regularly under the microscope if the spore is released, and then we take off the segment and then put it in another container to let it to continue to release. And then it took about like 5 months rearing them in the lab condition. And then after 5 months, we brought it into the hatchery condition. And to complete the life cycle, the F1 generation, that is not yet the complete life cycle, that is from tetrasporophyte to gametophyte, that the gametophyte becomes fertile, that was about 10 to 11 months. And then the F1 generation, we also induced them to release, and then we are completing that life cycle now. So the next generation is now in the lab and waiting to be transferred to the hatchery. So
thanks for the explanations. Yeah, and one more questions from our audience asking about what conditions are missing in the most commonly cultivars strains, which disable them from either sexual or asexual reproduction through sporogenesis. And is it their reproductive structures are entirely absent, or do they sometimes form during cultivations?
Okay, so that's an interesting question, no? Because most of the cultivated cultivars now, we don't usually see reproduction, whether they are gametophytes or tetrasporophytes, that we do not know. But there was a report from De Paula that they imported seaweed from the Philippines, and it turned out to be tetrasporophyte, and then they were able to induce tetraspore release and then grow them until a certain stage. So whether what we are cultivating now is tetrasporophyte, and whether there are farmers looking into the cross section, because it's not easy to see the tetrasporangal nematicia, no? Or whether there are scientists continually doing some sampling and doing some cross section to look at the tetrasporangia, I do not know. So because it was easy for us to detect reproductive structure in the hatchery condition. And in the hatchery condition, we're just using 70-liter aquaria, growing the progeny there. And after 7 months, they become fertile. So we actually have a male gametophyte, a female gametophyte, and a tetrasporophyte in separate tanks, because eventually we separate them, each individual, each life stage, we separate them into different tanks. And then we see reproductive structures. How come we don't see this in the field? I do not know. Yeah, maybe we need to do more observation, or maybe because the plant in the field is continuously asexually propagated for maximum about 60 days, and within 60 days, they might not be able to produce the reproductive structure. Like, for example, in our case, it took about 7 months before we were able to see reproductive structures. So if we could have a setup in the field, cultivated strain, and then just grow them to 7 months without harvesting, I do not know if we will be able to find some reproductive structures, if we will be able to establish if the farm cultivar is a tetrasporophyte or a gametophyte.
Thank you very much for the valuable insights, Dr. Roleda, and also for the detailed elaborations of the questions. And now from the Philippines, you are going to move to Australia to discuss with Dr. Zanny Langford.
Okay, thank you very much.
It is great to meet you. Thank you, Dr. Roleda. Dr. Langford.
Hi.
Zanny. Hi,
Yes.
it's great to meet you here.
Yeah, hi, good to see you.
Yeah, and the title of the presentations is Remote Sensing of Ecomatoid Productions. And like other panelists, we managed to collect some questions related to your pre-recorded presentations. One of them is, will you be able to use multispectral cameras and scan for the disease and or for poor growth?
So this paper was a collaboration which was part of our previous work that we'd done using Planet Labs imagery, with some new work that we're doing with some of our colleagues looking at automating that process and other sources of imagery. So my co-author for that, Swanti, is also on the line. So she's better placed to speak about the technical aspects of the process, whereas I'm more looking at the social and economic aspects. So are you there, Swandy?
Yeah, I'm here. Hello. Yeah, so multispectral. So I was looking at using satellite imagery, and what I found actually was that using NDVI, which is a vegetation index, so which is basically a ratio of the red and near infrared band. Actually, using that, I was able to do quite good segmentation of seaweed beds. And so, yes, definitely multispectral cameras, I think, is very useful to doing the segmentation task. I don't really know much about seaweed, so I don't really know like any sort of physiological aspects to this, but just from the imagery point of view, the multispectral is important.
Okay, yeah, thank you. And one more question that perhaps you both can elaborate. Our participant asked how much more detail can you get or can you obtain in discerning the crops?
In the what, sorry?
Detail. How much detail that you can get?
Well, it's limited to the scale of the imagery, so not a lot of detail. It's sort of good enough to get an approximate idea of the scale of production and how that varies between seasons and between years.
Mm-hmm.
But it's definitely not at the same level that you would get from drone imagery. So using this data, I guess it's really important to combine it with other sources of information and data collected on the ground and other types of data if we're looking to get better accuracy, but also to make sure that what we're sort of seeing is appropriate and to sort of ground truth the satellite analysis that we're doing.
If I can just jump in there.
Okay.
The satellite images are, so Sentinel-2 is on a 5-day basis, so we can actually get data on a weekly basis as long as the cloud cover is, as long as there's not so much cloud cover. And there's also planet data, which Zani was looking at on a monthly level, but they also have just daily images, which are at a 3 meter resolution. So we can use that as well to get data at a daily level. But what I found was that earlier in the season, earlier in the year, there's a lot of cloud cover. So more work has to be done on doing cloud masking to get data from the early months.
Yeah, and thank you. One more questions is what do you see as the future of imaging related to phyconomy in determining seaweed growth and health?
I think that there's quite a lot of opportunity for using that data. I think at the moment, the data, at least in Indonesia, that's produced officially is not very accurate. So I think there is a lot of scope to improve the accuracy of data collection. I think that could have wide-ranging implications for a range of different actors in the value chain to help improve coordination of the industry. And it can help with planning all sorts of things from distribution of seeds to drying or collecting, a better understanding of where seaweed is being produced and when could, I think, be used by a large number of people.
Okay, thank you very much, Dr. Zani, for this. And due to the time considerations, we would like to move to the next panelist, Dr. Anisha Hurtado and Dr. Alan Critchley. And we also have several questions from the audience.
Yeah,
Dr.
I'm here.
Anisha and Dr. Alan, yes?
I'm here.
Okay. Yes, thank you. The first question is, what do you think the key modes of actions might be? Have these been investigated to the same levels as impact of phycostimulants on land plants? And if not, what needs to be done? And are there any collaborators in the audience who might be interested in this aspect?
A very interesting question. In fact, I really prepared the response for this because firstly, seaweed extract biostimulant has been used for how many decades in agricultural crops, and it's only lately in 2009 that it's being used in seaweed cultivation, primarily for eumatoids. But now we have other seaweeds. So the first thing that we were looking into is, of course, the acceleration or hastening of the growth rate, and then will this have an effect on the incidences or the occurrences of disease and pest infestations? So with our several experiments in my team, not only in the Philippines, but also in Malaysia and also Brazil, we have found out that using the seaweed extract biostimulant has really accelerated growth rate and reduced the incidence of ice in particular, and even epiphytes and the phytes. Now the question is, what is the mode or pathway of this seaweed extract in terms of the efficacy on the reduction of diseases and pests? That is the only missing link now to our studies. So I have been looking for partners to work with me and to work with Alan and me also, and the rest of the team in Southeast Asia, especially Malaysia, to this effect. And I welcome collaborations, and I'm open for it.
That's great to hear, Dr. Anisha. And at the more technical level, one participants asked he found that it appears micropropagations result in good propagules. And it would be interesting to know how fast the scale up is, especially with AMPEP. How much time do you think would it take for a kilo of seaweed to yield, for example, 100 kilos of microphagol material?
Yeah, again, and a very interesting question. And I was really challenged because based again from our personal experiences with my team here in the Philippines and in other countries, it takes around, okay, let me give you a very short span for the whole propagation, that it takes only 30 to 45 days in the lob, and then we bring them in the land-based hatchery. If in case the weather in the sea doesn't warrant cultivation, so we keep them for the transition period, and then after that, another 30 to 45 days, we bring them to the sea, and then afterwards, 30 days, and afterwards, wow, we will be going there for the line cultivation. So practically, we can do it in 4 to 5 months. Ready for, we have already cultivars, or rather seedlings for cultivation. And your question is how many days will it take 1 kilogram to reach 100 kilos? Okay, so I base this answer from the growth rate, that assuming that you will maintain a 7.8% per day of growth rate, then 1 kilogram will reach 100 kilograms in 90 days, equivalent to 2 growth cycles of 45 days each.
Thank you, Dr. Anisha. And one last questions for you and Dr. Alan. Our participants said and asked, the AMPEP is derived from the brown seaweed as Cophyllum. What is your opinion of extracts made from other seaweeds? How do you think about this?
Yeah, of course, if the participant listened to my concluding remarks in my presentation, it is not only Ascophyllum nodosum that can be sourced as a seaweed extract. We have other brown seaweeds in the temperate countries like Eclonia, Macrocystis, Saccharina ondaria, and in the tropical countries, we have the Sargassum, we have the Turbinaria. However, these are not yet tried commercially, or probably they have been tried, but not that into a big scale. So there's no harm. There's no harm in trying other sources of seaweeds. Actually, when I first used this Ascophyllum, I studied the amino acid content of this Ascophyllum seaweed extract powder, and I found that it has a lot of amino acids. And I tried it first in 2008 on the micropropagation, because I have been previously, since 2000, I have been using the PES, the SS over 2, or even the Von Stotts as my culture medium for tissue culture, but those are really very expensive. So I tried using this seaweed extract from Ascophyllum plus PGR only, and it worked. I was able to generate direct axis plants of Capophycus of different strains already. So we published that paper, and now other countries are following. So it's not that Ascophyllum is the only source of seaweed extract for micropropagation. There are a lot of brown seaweeds, perhaps even the red seaweeds or the green seaweeds, to be tapped and explored for purposes of micropropagation.
That seems promising. And thank you very much, Dr. Anisha. There are still many questions actually for you both, but due to limited time, we are going to invite our next panelist, Dr. Nesh. And
Okay,
Dr.
thank
Nesh's
you.
presentations, thank you, is the seaweed industry development and lesson learned from Indonesia to the world. And there are several questions already offered by our participants. And one of them is in relations to the stakeholders. The world, of course, need more seaweeds, not only for the ecosystem function and food, but also to help to complete the United Nations SDGs. You have provided valuable insights in your presentations into what next. How do you think these stakeholders and producers are they listening, or will they listen? And if not, could you please predict what are the good, the bad, and ugly scenarios for the future? Dr. Nesh.
Okay. Well, number one, before I answer that, let me say that our company, C6, is making extracts like Ann was just talking about from Capophycus. So we have a factory in Bali that is making that, and it's already an established product. Okay, to answer the question you just asked, I think I addressed this in the section about value chain governance. And to me, the way forward is relational value chains, where all levels of the value chain are collaborating with each other on developing new products, on implementing sort of biosecurity measures and all the rest of it. In other words, under a relational value chain governance, all value chain levels are talking to each other and collaborating with each other. That is the key. That pulls in all stakeholders.
And one question was also raised in relation to a lesson learned in Indonesian region. As we all know, Indonesia is a large country with many different islands and different circumstances. How do you think the similar development can be expected to be implemented or scale up in other regions within Indonesia or even within Southeast Asia region?
Well, I think lesson 9 had to do with the coral triangle. And I think that, you know, there is a just massive development potential in the whole coral triangle. In Indonesia, we are blessed because we do not have typhoons and we do not have wars going on. Those are two things that make it possible for people to go anywhere in Indonesia and connect value chains. As you go further east to Papua New Guinea or even into Papua, Indonesia, Solomon Islands and so forth, I think there's huge undiscovered potential. And even in East Indonesia, I have lived in East Indonesia, in Maluku, and in Ambon, and in Tuau. And there's just a mind-boggling opportunity in that whole area. So Indonesia, in particular, the Coral Triangle in general, in my opinion, has the capacity to increase seaweed production by an order of magnitude over what it is now.
Thank you, Dr. Nesh, for these valuable insights and also for answering the questions. There are still many questions for you actually, but unfortunately, we are now preparing for the last sessions. So I would like to provide a brief summary to our sessions. I think we have learned and discussed several good practices from several countries in Southeast Asia and also Australia and Latin America, such as Chile, and also some latest development offered by our colleagues from UK and French. And we can see some opportunities for further collaborations to further develop this tropical phyconomy. And we are very pleased to enjoy both of your pre-recorded presentations and your responses to the questions. Once again, thank you very much for your time, and we apologize to all participants who offer us some questions, but we do not have enough time to respond to their questions. And I would like to invite my colleague, Dr. Inaya, to also provide some concluding remarks. Dr. Inaya.
Thank you, Sudirman. Time on my on the screen, we still have 12 minutes to go. So I think we still have, there's at least 2 questions that come during the sessions, and I found it's very interesting. Maybe we can go for that if it's okay. We still have 12
yes, please.
to go. Okay. Yeah. Can I just go for it? The question is go to Dr. Anicia and for Simon. This is from India. I found some Capophycus species tissue culture didn't growing well in some production areas. It's due to the transition of season from rainy to dry season. It's not growing well during the dry season. How to minimize the risk? The tissue culture could not grow well after 3 crop cycles. How do you think, Dr. Anne Hurtado and Simon Davis? Go for Dr. Anicia first, and then later on Simon. If not, I think we still have time for that.
Okay, I'll go for the last question, or I would like to react on that. The tissue culture, the Capophycus is only good for 3 growth cycles. Yes, I agree with you, because we have already some experiences in the Philippines, especially at Sif Deck, where we outplanted some in a seaweed farm here in Iloilo, and after 3 crops, they didn't grow as much as the first and the second growth cycles. So I think that's one of the advantages of having the land-based nursery, wherein you have to while the temperature in the sea is not favorable for field cultivation, then you have to keep your seaweeds in the tanks so that you will be expecting positive results if you bring them out, because it's not enough that if you have already some plantlets in the lab or in the land-based hatchery that you have to bring them out. You have to synchronize it with the environmental conditions of the sea, because for one thing sure, seaweeds grow much faster in the open sea than in the lab. So you have to be your weather conditions should be synchronized with your programming in the production of your plantlets, because if you don't do this, you will just be having an exercise in futility, and your plantlets will just put into waste. So you have to make a program on how to operate from the lab to the sea, to the land base, and to the sea base, or even into the field cultivation. Otherwise, and you have to really synchronize it with your local weather conditions to minimize high risk.
Okay, thank you, Dr. Anicia. Simon,
welcome.
Please.
Yeah, thank you very much. I think I'd like to start where there's a couple of questions that I have really before I could give a thorough answer. And the first is one that Dr. Reddy's listed in the chat is, when we talk about tissue culture, is that from callus or is that from micropropagation? Because there is a lot of confusion in conversations, and they're very, very different types of technologies. And I think I agree with Dr. Anisha, you know, environmental factors, seasons are crucial, but it would be worth zooming in a bit and discovering, is that declining superiority after 3 cycles, is that due to a certain fatigue, a declining advantage of the tissue cultured seedling? Or is it due to a mismatch in the season? It was previous in an optimum season, and then it's just in a less optimum season. So I've got more questions than answers, I'm afraid.
Simon, can I answer you with your question?
Yeah, go for it.
Yeah,
go for it.
actually, I gave that example here in the Philippines for three growth cycles, which is the something like short period, but they use a different, not seaweed extract as the culture medium, but what my collaborator or my partner in Malaysia is using is purely seaweed extract and a little only of the PGRs. And until now, we started doing it in Sabah and Puana 2018, and then it still continues up to the present. So the same protocol we are using. So far, so good.
Yeah, fantastic. So that was micropropagation, correct?
Yeah, actually there are two prongs in the micropropagation using culture techniques. You have the callus, from the callus, and from the direct access formation. So gotcha. We use the direct because it's shorter in time. In a shorter period of time, you have already new plantlets, because callus are undifferentiated cells that it has to transform into differentiated cells to generate plants, the whole plant.
Yep.
Yeah, absolutely. And I think, thank you, Dr. Anisha, for this. And in our dialogues, I think we should really be more specific. Are we talking about from callus, or are we talking about from micropropagation? Because in communications, there's been a lot of confusion. And I would just suggest that for communication in the industry and academia, about being a bit more specific that way.
Okay, thank you very much, both of you. Thank you. And we still have 6 minutes to go. Hopefully we can do effectively. There's one question for Dr. Lim. Dr. Lim, are you still here? Hello? Dr. Lim, there's one question.
Professor Lim?
Yeah, I'm here.
Yeah, Professor Lim.
Okay.
Okay. Yeah, there's one question. Hopefully you can answer it in 5 minutes. Are the available dendrograms or haplotype network, which include worldwide achomatoid specimens, could infer biological patterns other than taxonomic or biogeographic pattern?
Okay,
Please, your
thank
comment.
you for the question. Actually, I already answered in the chat box, but it's okay.
Oh, okay. There's another one question if it's, but your comment, please.
Okay, so as regards about the haplotype network is actually just to review the genetic composition of the strain that is available. So it's not really for looking into phylogenetics or taxonomy. Yeah, I hope I answered the question. Yeah.
Okay. There's there's another one I didn't follow. Is it already answered too? It's about the sorry, what is it? It's about the can we improve the vigor of Kappa ficus through polyploidy techniques? Your comment, please. Is it already?
Can you repeat your question? I didn't get.
Yeah. Can we improve the vigor of Kappa ficus through polyploidy techniques?
I think at the moment we do not have any data for euchematoid, so we do not carry out the experiment. So I think it's very hard for us to comment at this stage of time. We need experiment to look into that. Yeah.
Okay, thank you, Professor Peck. Thank you for all panelists. Thank you, everyone. We finally come to the end of the session 3. At the moment, we will now watching the video on opportunity for collaboration on seaweed safety. After the video, we hope that you can join us for the session 4 later at 4:15 in Makassar time. I think that's it for the session 3. Thank you very much for everybody, for all the panelists, for all the participants. Me as a facilitator has to say thank you and see you in session 4.
Thank you.